Moving on, our third session is going to be taken by Professor Utpal Tatu. So we welcome you, Professor, and yeah. So he is a molecular biologist, a biochemist, and a professor at the Department of Biochemistry of the Indian Institute of Science. We're very grateful that you're going to be teaching us about the global access to vaccines, and we're looking forward to, our fears regarding the vaccine being addressed by you. Thank you. Sir, you're on mute. Could you please unmute yourself?
Okay, good. Good morning, everybody. Very happy to be here today talking to the students. I think large number of school students, I understand, and very, very, very happy to share some of my experiences and ideas about the topic that we are deliberating today. Thank you for your kind introduction. It's a pleasure for me to actually be at this forum. First time for me, but I think it sounds very exciting. I also heard the talk earlier this morning, and very nice to see the question answer sessions and interactions that students are having with the speakers. I'm going to share my screen briefly, but what I'll do is, I was given the topic of talking about vaccines. About global vaccine availability and so on, and which is what I will definitely cover that part in my discussion, in my talk in the next few minutes. But I thought what might be also exciting is to share with the students what can a scientist do? How exciting is it to be a scientist? And what are scientists' responsibilities towards society? And through my personal experiences, I'll narrate what I have done, a little bit about what I've done, and also about how what an exciting opportunity it is to be doing science, to also be a science student. You really don't need to be actually a scientist, but even if you are a science student, how exciting it is actually, and then how important it is to be able to think, what can you do when the world is facing a challenge of the kind that we are witnessing today, the pandemic. Every science student has an opportunity to do something. You really don't need to be a scientist. And then what are the possible things that you can do through your ideas, through communication, and at the end, and of course, how does it, what kind of opportunities does it provide for students to interact with the society during this time? This is what my hope is that I'll convey in the 30 minutes or so that I have. But I will share with you some of the things that I have done, and then what are other scientists also trying to do while we talk about the vaccines as well. So yes, I'm going to put the vaccine story in the context of the title that I put forward for you here. Role of a scientist during the pandemic, and then COVID-19 research, diagnosis, and vaccine. Touch upon all of these aspects for the students, and I will try to make it more interactive and in a way that we I hope I can convey to the students the excitements of science, and possibly also trigger some of the students to take up science as a subject to study further, and then take it forward as a career. So let me start with this very simple idea. Today, of course, the pandemic is staring at us, and we continue to engage with it, understand how to deal with it, and so on. My own interactions as a scientist have been in the area of neglected infectious diseases. So this is the term that I want to spend a couple of slides talking about: neglected infectious diseases. And I also want to take back this discussion to the time when I decided to do science, and how I decided to do the topics that I research on. And again, my intention is that some of the students may get may get interested in pursuing science after the school, college, or whatever they are doing at the moment. So neglected infectious diseases are the kinds of infections that are not heavily researched upon. And COVID-19 in some ways is an example of that. There are large numbers in all over the world where infectious diseases take a toll on various economies, various populations all over the world. But among them, there are diseases which are predominantly present in underdeveloped and developing nations. And those are the diseases which do not often get enough attention for the reason that there is no financial benefit in, for example, developing diagnostics, developing drugs against such diseases, because they are in poor income category of nations where drugs and diagnostic kits and vaccines will have to be at a very affordable price. And so pharma companies often don't see that as a encouragement. That's not where their money will be returned or their investment can find return. And justifiably so. Of course, pharma companies have to think about their business and their sustenance. So it's understandable. But that's really why many diseases remain neglected. They're not enough. There's not enough research happening on it, and there's not products coming out to manage these diseases. And the list of these diseases is large, of course. But what I've shown you on this colorful slide is some of the infectious organisms, such as amoebiasis, giardiasis, trichomonosis, plasmodium. It's no longer neglected, but it was on the list of neglected at some point in time. And then many other infections. And these are the infections that my laboratory at Indian Institute of Science tries to research on and tries to understand what can we do, understand not only in the lab, but also in the field. So that's really what neglected infectious diseases are. And when I started my research career after finishing my studies and postdoctoral studies, my motivation was really to look around in the society. What are the current problems that, as a scientist, as a biologist, I could make a difference with? And that's really how I decided that diseases which I saw around me at that time, which was malaria and trypanosomosis and diarrhea, I decided that can I do something on it? I really didn't have much background in studying these infections earlier, but I decided that this is how I will interact with the society. So that's really how my journey began in this field. So what I'm going to do in my talk in the next few minutes is to first start giving you a broader perspective about neglected diseases, its connection with our environment, our animals, and us, ourselves, our activities. That's a broader topic which results in pandemics of the kind that we are witnessing today. I already talked to you about neglected diseases. I'll dwell upon it a little bit more and then jump into the main topic of my own experiences in COVID-19 and then eventually a little bit broader discussions on vaccines and the way forward with where we are heading. Pandemic is not over. Everybody's still worried about what might happen next. So this is the part that connects with what I told you earlier on, that neglected infectious diseases. And then the idea that I'm trying to convey through the slide that I've shown you here is that majority of diseases that we hear about today, in fact, are contracted by humans through its environment. And large number of them, in fact, as many as 70% of our infections actually come from animals. And this is an important point that I want to get across to you, that we interact with our animals very closely, and our interactions with our animals is changing rapidly. Be it agricultural animals, cattle, sheep, buffaloes, be it recreational animals, be it pets, all of these have an impact on, and while we think about humans should remain healthy, but we often forget that the health of humans actually depends on what is around us. It's equally important to keep our animals healthy in order for us to be healthy. This is what I've been preaching and practicing for the last more than 20 years that I've been here and doing my research. And then, of course, last 2 years have been what an eye-opener it has been. I think many of the things that we talk about has unfortunately come true. That because of the neglect of dealing with animals and taking care of and examining animals, infections have jumped species boundaries, have come from animals to humans. And I want to show you here that large number of diseases, and the lists are here. I'm not sure if you'll be able to read it, but rabies is an example that all of us are aware of, and many other viral and non-viral parasitic infections coming from small rodents to birds to deers to pigs. We know about swine influenza from cattle, and then of course poultry. All of this, of course. And then, of course, there is the environment, which includes the flora and fauna, which all are potentially source of transmission of diseases for all of us. And that's something that I want to get across. My own research has predominantly focused on such infections that jump from animals into humans, and the list of which I showed you early on. These infections early on, of course, are within animals, but then they transmit into humans, and then they can be transmitted from humans back to animals, and then among animals, and as a result, they become they stabilize in the environment. It's very hard to then deal with. As I talk about this idea, I want to put forward this simple table. If I ask, if this were to be an offline seminar, I would ask my students that tell me what are the most common infections that you've heard of, and the answers would be influenza. People would talk about Ebola because they talk about it in newspapers, and would talk about, of course, things like Zika virus and Nipah. All of these are there in the table that I'm showing you here. Ebola, Zika, Nipah, swine, severe acute respiratory syndrome, Middle East respiratory syndrome, avian influenza, and then most importantly, in recent one, the Wuhan coronavirus outbreak. The common thing that ties all of this is that all of them, as I was telling you earlier, have jumped from animals and others, mammals, into humans, and sometimes from birds, as in case of avian influenza. I will not go deep into the science of it, but all of these infections, be it most of them from viruses, actually have caused havoc. In many of them, there were outbreaks, major outbreaks, a lot of fatalities happened. Many of them were controlled, and then we are still dealing with the last one, which is Wuhan outbreak, which originated in China. And this particular part of the table talks about where these infections originated. So yes, indeed, what I'm telling you is a reality. Infections are spreading across very efficiently all over the world. So what I'll tell you that as a scientist, then what have I been able to do about it? You know, as scientists, oftentimes we do research in our laboratories, and that's what I do. And we do fundamental discoveries by observing organisms, by studying them in the laboratory, observing them under the microscope, and applying whatever scientific tools there are to understand how these organisms work, how these infections spread. And then one publishes academic papers to share with the academic community, wherein collaborations can happen, exchange of ideas can happen. And that's really what life of a scientist is all about. It's all about being able to make observations that excite you, and then share it with the scientific community. That's really what one does as a fundamental scientist, which is what I started my career as. Over the years, then I realized that many of the diseases that I'm dealing with actually cause great damage in the society. And so I decided that, yes, I have to turn my attention away from the laboratory, also look at the surroundings and clinical samples, diseases as they happen in the field. And which is what I got busy doing in the last 10 to 15 years, applying or extrapolating observations from the basic science, laboratory-based research into the clinical. And eventually then, one hopes that many of these observations will result in some interventions, be it at the diagnostic tool level or drug level. And this is something that I'm embarking on. And in some small, minute way, we had some success. And again, I must say that this is not to glorify my small success stories, but really to make an example out of it for students who may find it exciting that, hey, this is the career I want to take off. This is what I want to do with my education in the years to come. So what I did early on is to be able to connect the laboratory science to the science as is applied in the field, to the problems that are faced by people, by farmers, by animal owners, and also by community at large. I created a platform, and my institute, Indian Institute of Science, was very, very supportive and helping us create a platform where we could actually convey our science to the society. We could actually create products. We could actually provide service at a very different levels. They, in fact, gave me access to a laboratory where all of this interaction can happen in a way that there would be no limitation in terms of communicating and taking science out of the laboratory. And that's what resulted in a possibility where observations made in the laboratory could then be converted into interventions for society at large. I'm going to jump into COVID-19 because that's what many of you are waiting to hear. But before I do that, I want to make a simple example. Since I gave you an example of a neglected infectious disease and how animals are transmitting diseases and how our health actually depend on the environment, one of the most common example I want to raise. Many of you have heard about rabies. While today we are in the midst of coronavirus pandemic, but we must keep our eyes open and understand that there are many other infections which have caused damage to us, and they continue to cause damage to us. And rabies being an example, again, a zoonotic infection transmitted from animals to humans. We do not have effective way of diagnosing such infectious diseases even today. And India is actually an important place where a significant number of rabies cases are reported. One of the highest numbers world over of rabies, and rabies is considered to be 100% fatal. That means any human which contracts rabies is sure to die, unfortunately, and as a result, an important disease. So just a perspective that we are dealing with pandemic like COVID today, but there are host of other infections that we still do not know how to deal with and could become bigger threats in years to come. And this is something that we started working on and developed very effective method of diagnosing this infection. Infections like rabies, of course, spread through animal bite, such as dog bite, to human, and then the virus enters into the bloodstream, finds its way through the axon, to the neurons, to the cerebral, to the brain, and that's how it causes neurological diseases, and eventually the human dies. But there is no definitive way, actually, to diagnose this infection at an early stage. Even though the person who contracts this disease will die, it's important to isolate this person, especially the animal, away after identifying that it's infected, in order that the spread of the infection can be prevented. And that's really why diagnosis is very important. And that's really what we did. We developed a method for early detection of rabies. Even when there are very early signs, we don't yet fully know that the symptoms are there, but we can actually detect the disease. And that's really what was lacking. But what I want to show at a broader level is that, like rabies, there are many other infections transmitted by all kinds of animals. And in India, there have been outbreaks at various points in various parts of the country in this context. Many of us are animal lovers. And that's why it's very important for us to understand that when you take care of our animals, this is an important part, to make sure that they do not get infected, and then further on, they do not transmit the infection to animals. COVID-19 is a glaring example of an infection that was spread from the wet market in Wuhan, where it's an animal market where people sell animals of variety of kinds, including, in this case, of course, bats. Bats typically is not a food for humans, but some people they like to eat bats. And as a result, this is how the early transmission may have happened. We don't know exactly what, but I think there are a variety of stories. But that's really the context in which I'm putting this emphasis on taking care of animals and understanding zoonotic infections, that is, infections that are spread from animals to humans. In the next few minutes, I want to tell you again how important it is to think about it in a broader perspective. I was myself doing a lot of work on coronavirus infections in animals for last several years. So to tell you that while we know that this infection is probably spread from bats to humans, in fact, coronavirus very common in dogs, cats, and many other animals. And for the last several years, my laboratory was actually doing diagnosis of coronavirus infections, even before the world had heard of COVID-19. Even before the pandemic struck us, coronavirus infections continued, and then there were diagnostics that were already available for animals, and we were getting routine samples from dogs and cats for diagnosis of coronavirus. As a result, already in the last few years, we had very robust methods available for diagnosis of coronavirus infections in animals. I want to tell you, some of you already know this, and previous speakers would have talked about it, that coronavirus derives its name from crown-like appearance because of spike glycoproteins on the surface. This is how the virus looked, be it COVID-19, which has infected humans, or COVID or coronavirus infection that happens in feline, that is in cats or in dogs. They look the same. They are the same, almost the same. At genetic level, they are very closely related. So what I would like to emphasize is that because the awareness that we had, that animals can transmit infections, we actually were at the right place at the right time in developing effective strategies for mitigating coronavirus infections. And as you all know, coronavirus COVID-19 diagnosis depended a lot on RT-PCR methods, which is what, of course, we were doing even before COVID-19, and that made us, made it possible for us actually to create a test which was very effective even for COVID-19. I want to show you this. It's a bit of a scientific diagram for school students. Some of you may understand. It's a kind of a phylogenetic tree, which tells you the relatedness of different forms of viruses, and that happens through genome sequences. For example, the COVID-19 that infects COVID virus that infects humans is closely related to the bat, but also not so far from those that infect horses, those that infect cows, those that infect the cats, or from porks or to the dogs. And as a result, there is a lot of exchange of information that's possible. And we, of course, were working on the domains other than humans even before COVID-19, and that helps us to actually pivot in the right time and apply technologies to develop products. Very quickly, I'm going to tell you that early on, we did some very, very interesting lab-based observations. In February, March of last year, when COVID was just hitting India, we started taking clinical samples and doing genome sequencing. And that gave rise to understanding about what kinds of sequence changes, be it genome sequence changes or amino acid substitutions that have happened in the virus that's coming to India. And we found very quickly that the virus that we found were closely related to those from Bangladesh, and then those in turn were closely related to the to the European form of COVID-19. Well, I'm going to go a bit fast and tell you that we used this knowledge early on and created a platform where we began to discuss with international bodies. In fact, in February of 2019, when the story was just beginning, we had a global sort of a conference in my laboratory where coincidentally there were scientists from all over the world were anywhere supposed to visit who were experts in infectious diseases. And that's really how on 28th January, we set up a protocol because Wuhan cases were just being reported. And then, of course, as a scientist, we set up a protocol to see how the disease would progress, not knowing what was going to happen. And then, of course, interacted with the government and eventually developed robust methods to mitigate this pandemic. One of the early collaborators that we had was many scientists from within the city. This is February, actually end of January 2019, when a scientist from NIMHANS is giving a talk at Indian Institute of Science, along with few of us who put together a team on infectious disease to understand what we can do in this scenario. We interacted closely with government of Karnataka and created a strategy to understand how we can control the disease. I'm going to skip the slides, but to tell you that this allowed us to create a robust technology for creating a way to detect the disease at the right time. And this was important because now as we look back we have so many different tests, and RT-PCR is a common household term everybody understands. But at that time, we had to think about how to make how to train people to even do RT-PCR at the scale that was required at the national level. And some of it, actually, I had a chance to contribute. And in turn, we developed a test ourselves for COVID-19. What all of this resulted was actually a decrease in the number of cases because large number of tests were being conducted all over India. ICMR did a wonderful job in creating centers for testing. And as a result, today, of course, we are at a stage where disease is in control at a very, very good level in India in a very enviable manner. What our country has done is exceptional in terms of mitigating the disease. Of course, the risks are still ahead and we need to continue to monitor, but yes, we are in a good position. With this sort of a general understanding about how science has helped and how as a scientist I have done my part in interacting with the society, I will jump into some additional aspects of vaccines and the way forward. Now, students, of course, are aware of the term vaccine, but the term actually was devised by a medical doctor by name Edward Jenner, who talked about, who created one of the first vaccines. And the term came from variole vaccinae. It's actually something that's related to the smallpox of cow. And that's how the term vaccine came about. And that's often interesting for us to think because as students, we are always, confronted with various complicated terms and we accept these terms. But it's interesting for us to think about how has this term originated, because lies behind that term is an interesting story about how the whole idea would have come up. Who was the person who thought about this term? Who was the person who conceived this idea that there would be a way of preventing an infection through now what we call as vaccine? And that gentleman was Edward Jenner. The story that I narrated to you in the first half of my talk ties up very well with the discovery of vaccine. Edward Jenner was, of course, a medical student and then a doctor. Actually, in the whole idea about vaccines generated because Edward Jenner was working in the field. He was working actually close to an animal farm. And he observed and this is, of course, way back in 1700, actually, he was working as an apprentice, a very young doctor who was first appointed to be in the field. And he made a simple observation. And I would require students who are listening to this talk to pay particular attention to what I'm saying, because the idea is very important. It's very important to ask this question that who would have thought of this idea first? Now that people are developing vaccines and newer vaccines will get developed, but who and how was that idea actually originated? So really, Edward Jenner, as he was working in the field, he realized that there was a disease which we all know about, smallpox. The new generation doesn't have to deal with it as much because it's one of the first infections to have got eliminated from the world. But smallpox was a common infection. In 1700, 1800, smallpox was devastating. It of course was also disfiguring because it caused pustules all over the body. But that's where the virus was present and there was no solution for it. But Edward Jenner, as a doctor, went around and he made an interesting observation. And this is what is written here, that Jenner learned that in some common rural areas, dairy workers would never have the often fatal or disfiguring smallpox disease. This is really how it all began, because he was working in the field and he heard through community gossips that this particular community or actually rural folks, people who work with animals, people who work with cows, somehow do not get smallpox. This is really how the whole vaccine idea came about. And this is very important because we can, of course, while we keep reading books and we understand the basics of science, it's also important to connect that with what's happening around us. And this is a wonderful example. So Edward Jenner related this observation to everything that he had learned and thought that, how is it that this particular population doesn't get it? Then he realized, of course, that these animals that these people were working with, the cows, of course, get cowpox disease. And then he conceived this idea that possibly exposure to cowpox was happening to this rural folks, and somehow then some mechanism was getting triggered which prevented them to get smallpox. This is really how the idea was conceived. So if you sort of conjecture this particular piece of information, that how a person developed an original idea by connecting his knowledge with what was happening in the field, that really would be the biggest thing that you can actually do as a science student. I will move forward and tell you then rest of it is just basic information that he then, of course, thought of taking pus from the hand of a milkmaid with a cowpox disease, which was, and scratched it into the arm of an 8-year-old boy, and eventually found that this boy was then, of course, did not get smallpox, which was a related disease in humans. So cowpox exposure was preventing smallpox case. This really how then that's how he developed this into a vaccine and reported that his vaccine was safe in children and adults. That's really what I would call the first clinical trial of a vaccine. But I hope that you appreciate the story of how the whole idea came about. And then, of course, Jenner went on, of course, to make a full-fledged vaccine, which was accepted world over, and it controlled this infection. One of the first persons to get this vaccine was Phipps. And again, that's a story that's present all over the find it on Google very easily. But having said that, let me just say, then of course, this principle they will continue. And Louis Pasteur then took forward this idea and developed vaccine for the virus that I talked about, rabies virus, because that was, imagine, it was a problem in an ancient disease from 1700 to 1800 until today. In fact, as we are speaking, there are possibly people dying of rabies. So these are infections which have been around for centuries and centuries, and we still do not have solution to this. Yes, we are talking about COVID-19, but there are many other infections. And that's why I brought up rabies as an example. Even in today, in our own city, in Nimhans, there must be patients who have contracted rabies through a dog bite and who are possibly going to die in the days to come. And this is a virus for which the vaccine was developed already by Louis Pasteur. But of course, there are some gaps that we haven't been able to meet. Lastly, in the last few minutes that I have, I'm going to take this idea a little bit ahead and tell you a bit more about the COVID-19 vaccine. I talked to you in the last few minutes about Edward Jenner's observations, how he came about with the idea of vaccine, how he developed a vaccine for smallpox. Louis Pasteur talked about rabies virus, and again, there's a very interesting story behind it, which I would encourage you to look at. COVID-19, there was nothing that complicated. In some ways, the conceptually developing a COVID-19 vaccine was not an intellectual challenge. There's a little information that you'll be very surprised to hear, that for animals, there was already a vaccine for coronavirus. For last decade, actually, many animals are being vaccinated for coronavirus infection. So proof of principle was already there, that coronavirus infections can be controlled by vaccination in animals. So it really wasn't as if it's going to be intellectually difficult to create COVID-19. But COVID-19, of course, had different sequences, it had different genomes, so you had to make a vaccine for it. So what was the big challenge? The challenge was not the idea but the challenge was implementation, as is often the case for many things, many things which have to reach a society. So if you keep that in mind, then I think the next few minutes will become more interesting for you. Vaccines, of course, are of different kinds. Now you hear about COVID-19 vaccine in newspapers, RNA vaccine, DNA vaccine, and it's confusing for all of us. So let me make it simple for students to understand, and many of you would know this very well, that vaccines are of different kinds: live attenuated, inactivated, subunit vaccines, toxoids, conjugate vaccines, DNA vaccines already out there, influenza and herpes virus infection and hepatitis virus infection. There is a DNA vaccine for it. And then, of course, now people are talking about RNA vaccine. COVID-19 vaccines are available in all shapes and sizes. You have the DNA vaccine, you have the RNA vaccine, you also have the inactivated COVID-19 vaccine. And there are examples of all of these vaccines available. But if you look at it, traditionally, large number of vaccines have been in the first two category or first three categories: inactivated or live attenuated or subunit. I will not have time to go deep into the scientific aspects of it. I would urge students to read it if they are curious. The point that I want to make is that COVID-19 vaccine, what is available to us? I'm going to come to that in a minute. Now, globally, the vaccines are becoming available since January. Actually, there already some people got vaccinated in October and November, even in India, I must tell you. But of course, it became publicly available in January. Large number of vaccines have come about. I told you about smallpox in 1700, 1800, so rabies, and then whatnot. Over the years, we have large number of vaccines. And the reason I'm showing you this timeline of vaccine development, which continues in the next slide, is that majority of these vaccines have been the conventional, traditional types of either heat inactivated, live attenuated kinds of vaccines. Very few have been of the kind that are becoming popular in COVID-19. DNA vaccines and RNA vaccines. So in other words, we know relatively less about this new generation DNA and RNA vaccines. But not to say that they are not safe, but I think this is a relatively new concept. And if I'm not wrong, among RNA vaccines, COVID-19 is one of the first, and which is interesting and going to be, I think that's the vaccine that's being given world over quite efficiently. A spread of what's available for COVID-19 vaccine just for the students. Again, it's a very easy information that's available. The most important points that I want to tell you is that various companies all over the world have developed, be it RNA vaccine, DNA vaccine, or the attenuated form of COVID-19 vaccine. The point that I want to emphasize here, and very proudly enough, that India, of course, boasts of having its own vaccine. A company called Bharat Biotech developed a vaccine indigenously, completely indigenously of inactivated virus, and that's actually appearing highly. The clinical trials show that it is very effective. But in addition, of course, there are other vaccines being manufactured in India, which they were not designed in India, but they're manufactured in India. Some of you may have heard that India is the pharmacy of the world. The vaccine manufacturing capacity in India is one of the largest. And India is manufacturing some of the largest doses of COVID-19 vaccine. As a result, we are able to, in fact, export to many countries. We have already exported to neighboring countries that include Nepal, Bangladesh, Sri Lanka, but we continue to export it to other countries. And that's something that has made India very proud because of the large manufacturing capacity that India had. But importantly enough, very interesting to think about that Bharat Biotech developed its own indigenous vaccine, completely conceptualized in India and developed in India, and government of India backed it up very well. In fact, a few weeks ago, where Prime Minister himself got a dose of the vaccine developed by Bharat Biotech, and he's supposedly doing well, and this vaccine is very easily, commonly available. But what I want to show you here is that vaccines, the challenge in COVID-19 vaccine really was less of conceptual, but more of implementation, of manufacturing. This is the first time ever in the history of mankind that you would need a vaccine that probably will have to be given to every human being on the planet in the years to come. So up until now, there has never been a situation where you would require a vaccine in such large amounts. That really was the challenge. How can we ever cope with the demand of vaccine at such large amounts? And of course, more and more vaccines being available. Of course, it all started with giving vaccine first to the frontline workers and so on, and then to the senior citizens, and now gradually it's becoming available to the others, and there's a lot of information. But when you think about vaccine, I want to flash a couple of slides to intrigue the students that you really have to think about precisely what is there in the vaccine. When you talk about DNA vaccine, what is the piece of DNA out there in it? When you talk about RNA vaccine, as I'm showing you in this slide from Pfizer, what is the specific RNA sequence out there, is there in the vaccine? Please do think about the specifics. That's where the science and exciting information is. DNA vaccine, again, Covishield, and all of those are actually DNA vaccines in a proteoliposomal particle for its effective delivery into humans. But this is what I was telling you, that India is one of the largest producers of vaccines. I already gave you example of some of the companies that are producing vaccines, and there are more that are coming into picture, and that's going to be a good news, not just for us, but for the world over. Very quickly, I'm going to wrap up and say that, well, all well and good that vaccine is available, but how did we create a vaccine in 1 year? January 2019, we began to have this problem. January 2020, vaccine 2021, vaccine is available now to us in about 1 year. January 2020 is when the problem began. 2021 January is when vaccine is now available to a large number of people. What I want to share is that typically, traditionally, vaccines take large number of years to go through research, preclinical development, and clinical development. It almost takes 5 to 10 years. And then, of course, the implementation, execution, transfer process to manufacturing, and then delivery all takes another few years. This is how the trajectory is. But in case of COVID-19, because of the emergency, people have actually fast forwarded many of the regulatory steps, and a lot of government support has been available. As a result, unachievable, what was thought unachievable has been achieved. That vaccines are now available. Multiple vaccines are available. New vaccines are coming out, and it's all good news. I'm going to end very quickly and say that we will continue to need new vaccines. Of course, COVID-19 vaccine is available, but of course, there are many other vaccines, be it Zika, Ebola. Some are ready and others are in the pipeline, and the experience with COVID-19 has helped us and will help us. Bharat Biotech, India's first COVID-19 vaccine, indigenously manufactured and conceived, that is very important. And then the cocktail of vaccines from various other countries, and some of them being manufactured in India on large scale. I emphasize that again. And with that, I want to end by this simple comment that pandemic has taught us a lot of things. It's an exciting opportunity for students to think about science as a way forward, to study further science and maybe make a career out of it and make a difference in the society. And of course, COVID-19 is a situation where scientists, politicians, regulatory bodies have worked together to overcome this challenge. And I think overcoming the challenge is possibly something that is too early to say, but we have made a small difference. And to leave some time for students to interact, I will conclude at this point and would like to thank you for your patience here. Thank you so very much.
Thank you, Professor, for that presentation. We have a few questions for you. The first question is from Cindy Yao from Fuzhou Lakeside International School, China. And she is asking you, why is it that bats react differently to the coronavirus than humans do?
Well, those are the species differences. Many infections, many organisms many animals can harbor organisms without damage to themselves. But when the same organisms enter another species, it can cause infection. And there are multiple various reasons for it. Bats do not succumb to coronavirus infections as humans do. The difference is in the way the infection is caused and the way the infection transmits. So as a result, until about a few years ago, virus which was residing in bats had not encountered humans. But when it did, because of the kinds of interactions that have taken place between animals and humans, in this case bats and humans, it did find entry into the human system. And then suddenly it actually there were mutations that took place and it adapted to the system and became a virulent form. So yes, it's all about the genes of coronavirus. That's where the mysteries lie. But broadly speaking, it's commonplace knowledge that organisms that some animals are completely comfortable harboring can be extremely harmful for other animals. That's because of the species boundary in terms of infection and in terms of the invasiveness that the viruses and bacteria can exhibit in different biological systems. If I have to explain this in a deeper, it will be a long time. But I hope I have given you some small information about the question.
Yes, thank you, Professor. Earlier, you showed us some pictures of some of the meetings that took place between experts on COVID-19. So we have a question from Manya Singh for you, and she's asking if there is a set of communication systems between medical experts globally.
It's a very good question. Manya, that's a very pertinent question. And this has been the biggest challenge. To me, it continues to be a challenge. So I'm going to answer this question in a bit bigger way. I'll take a half a minute to answer this question, maybe 15, 20 seconds. But the point is that even today, actually, there is not enough communication internationally as far as COVID-19 is concerned. You know that there is great differences in the way different populations have reacted to COVID-19. While in US, it still continues to be on the rise. The much more severe infection COVID-19 has caused in that population, but in India, and we do not have a clear answer. And so really what you're pointing is very important, that there has to be a lot of collaboration among the different countries, scientists. We haven't seen that a whole lot in the last year or so. And that's really why this question is still open. It's an unanswered question, that why have different populations reacted differently to COVID-19? So your point is absolutely valid. There have to be better platforms. And that's something hopefully people have been so busy just fighting this infection within their own countries that they haven't had enough opportunity to think about collaborations. But yes, some collaborations are beginning to happen, supported by governments. Discussions are happening among scientists, and platforms are being created as we speak. But yes, not at the pace that was necessary. We can do better.
All right. Yeah, Professor Tatu, we have to say thank you so much for giving us your time of day and for helping us understand that there's in fact a production of vaccines at a mass scale going on in India, and that people do not have to be fearful, and that their time to get the vaccines are going to come soon, and that we are in fact capable of exporting vaccines to other countries. Thank you so much for giving us that understanding. Thank you once again for your presentation and all the knowledge that you shared with us.
Yeah, thank you for the opportunity. It was a pleasure. Thank you.